Hello everybody, welcome back. We're going to finish up our discussion on the Loci Archaeota today. Again, it will probably be a relatively short lecture. And then the reminder is to please read that very old Lowe's paper that's online regarding the discovery of the Archaea. That is the paper that we will walk through on Friday. Okay? Any questions as we get closer to the end of the semester? Okay. So we left off discussing the fact that these authors have isolated a bunch of genomic sequences that belong to this deep sea archaeal group. And some of the organisms described apparently have eukaryotic family proteins. And these are the so-called Loci-Archaeum. Shown in the purple here are the sort of the families of eukaryotic proteins. And none of the other archaea that have been discovered have these families of proteins at all. So therefore, it was interesting to note that these Loci-Archaea apparently were perhaps precursors evolutionarily to eukaryotes. All right, and I told you that these eukaryotic family proteins belong to a selected group of proteins whose functions in eukaryotes tend to be involved in producing a cytoskeleton as well as a membrane trafficking system or some mechanism to build membrane-bound organelles. So now we're going to start talking about what these proteins are actually doing, or what it's believed that these proteins are doing. The first family of proteins that were described in this organism are the so-called small GTPases. These small GTPases tend to be signaling proteins, and they work by binding to GTP or GDP. And when these proteins are bound by GTP, they become active. And most often, an active small GTPase recruits another protein to it. So by being GTP bound, these small GTPases now bind other proteins. If that small GTP protein becomes GDP bound due to an inherent GTPase activity, then that small GTPase loses its affinity for that so-called effector protein. Okay? So here is a small GTPase. In this case, this is a member of the so-called RAS subfamily of small GTPases. And the GTP bound state of the small GTPase is regulated by a number of other proteins. All right? So there are some receptors at the plasma membrane that can now signal a family of proteins. These are guanosine exchange factors. These exchange factors now place GTP into the active site of a small GTPase. So now we've made this RAS GTP bound, and then it can go on to bind additional proteins called effectors, and then go on to have some biological response. So in this way, these small GTPases are acting as signaling molecules from some environmental signal to affect some aspect of eukaryotic cell biology. And then to turn off the activity of these small GTPases, there are families of proteins called GAPs. These are GTPase activator proteins that now stimulate the GTPase activity of this GTPase. and then turn it to the GDP bound form, thus shutting it off. The GTP binding to these proteins is not used for energy, right? So we're not hydrolyzing GTP to gain energy. We're simply using it to change the overall shape of that GTPase to bind or not to bind proteins. If we look at the organisms that contain small GTPases, which are not very often found in bacteria, we note that a lot of archaea, so this whole y-axis, everything in gray essentially is archaea. Archaea do contain small GTPases, but it's 3, 1, 4. When we start getting to eukaryotic cells in blue, we now start to see a couple dozen, hundreds, for example. And this graph on the x-axis is essentially percent of the genome. So even though we have approximately 453 small GTPases relative to the size of our genome, that's only about 0.8% of our genome. But now let's look at Lokiarchaeum. Lokiarchaeum has 92, which is already way more, an order of magnitude more than what's normally found in archaea, and it comprises almost 2% of its genome. So when we graph this, you can see that we've placed Lokiarchaeum within basically eukaryotic cells in terms of their importance of these small GTPases. So it looks like that the Lokiarchaea have greatly expanded the number of small GTPases, perhaps to respond to other environmental stresses or signals. And it's possible that these authors determined that the small GTPases in Lokiarchaeum are actually ancestral to eukaryotic small GTPases. But that was not proven. Okay, so what about actins? Actins are the cytoskeletal proteins of eukaryotic cells. This describes the dynamics of the actin cytoskeleton in a typical eukaryotic cell, where actin monomers that are ATP bound can assemble into long filaments, as well as into these branched filaments. So we can have both actin cables, which are bundles of straight filaments, as well as branched actin patches. These branched actin patches tend to be important at the membranes, the plasma membranes of cells, where they can deform the membrane. For example, the processes which drive phagocytosis or endocytosis in a eukaryotic cell require branched actin. Now, forming this actin cytoskeleton utilizes a number of very conserved eukaryotic proteins, including members of the ARP2/3 complex, WASP family proteins, so on and so forth. So not only are actins required to actually maintain the structure, to help maintain the structure of a cell, it turns out that we can place force on the membrane to actually extend that plasma membrane. So when eukaryotic cells create these lamellipodia to move across a surface, actins are intimately involved in that process. I described you the requirement of actins for membrane traffic or phagocytosis. And it looks like Lokiarchaeum have relatives of these ARP2/3 or these ARP family proteins, which could suggest that not only does Lokiarchaeum have an actin-like protein it has the ability to manipulate its dynamics, thereby using actins potentially to, for example, change the structure of its membranes dynamically. It turns out that some of these other tacharchia also apparently have some of these actins or actin binding proteins. One of the other highly represented groups of these eukaryotic proteins in Lokiarchaea are the so-called escort family proteins. These escort family proteins belong to several multi-subunit complexes from 0 to 3, whose job it is to basically be recruited in an ordered fashion to membranes and then force the invagination of that membrane, okay, away from the cytoplasm. So if we imagine that this lipid bilayer membrane in green is the limiting edge of a membrane-bound organelle in a eukaryote, these proteins will actually assemble on the surface of that membrane and then eventually push that membrane into the lumen. So we're pushing these membranes inside of this membrane-bound organelle. This is a conserved process that all eukaryotes do. One of the primary reasons that an organism will do this is to enhance or to drive the turnover of membrane proteins, and especially these ubiquitinated proteins, which are marked for turnover by proteolytic degradation. There are members of these escort family proteins that bind to that cargo and then push it inside of degradative vesicles, essentially. All right? And again, this is a well-conserved protein family with homologs from, in this case, yeast, essentially, to man. So escorts are involved in cargo selection. They're actually involved in viral budding, too. That is, eukaryotic viruses are known to hijack the escort machineries to help them bud out of the cell. Protein traffic, that is the turnover of membrane proteins. At least one protein member, this conserved AAA ADPase, VPS4, is found in many other archaea, but it's usually found alone. Right? This is the ADPase that recycles the last step. So here's the escort 3 machinery, whose job it is really to polymerize and push these membranes into the organelle. This VPS4 ATPase essentially stops the process. It disassembles these filaments so that we can turn over all of this escort machinery. So VPS4 is actually well known and it's described in a lot of other archaea. However, Lokiarchaeum appear to have at least one member of escort family proteins from each of the numbered complexes, right? So whereas before, most other archaea only have VPS4, the Lokiarchaea have members, for example, for VPS25, VPS2, VPS20, and again, potentially have the ability to form these multi-subunit escort complexes. This is another major distinguishing factor from Lokiarchaea from the rest of the archaea. Okay. So this cartoon or this figure is meant to highlight the different families of proteins that were discovered in sort of the TAC superphyla of archaea, the Euryarchaeota, as well as the eukaryotes. And the authors try to understand how these families of proteins evolved in these organisms. So you can see that we had some start, in some ancestral ancestor that gained a bunch of protein functions. Those protein functions were maintained in the branch that eventually developed both eukaryotes and Lokiarchaeota. And every time you see a bunch of X's, you see that this evolutionary line has lost these protein activities. So Lokiarchaeota and the eukaryotes, at least through this analysis, are linked by protein function at some point distantly in time. And these are the different families of proteins that these organisms appear to contain that are very different than the rest of the archaea. Okay, so this work now summarized this new superphylum of archaea. These are the Asgard archaeota, or the Asgard archaea. And they have protein coding regions that are more similar to eukaryotic proteins than archaeal proteins. This is not apparently contamination, because when the authors looked at the sequences of those open reading frames that encoded for these eukaryotic proteins, they were always flanked by much more prokaryotic genes, all right? So it's not like this was a eukaryotic fragment that snuck its way into the overall mix of DNA. It apparently is part of these much more prokaryotic or archaeal genomes. And this was thought to support this so-called eocyte hypothesis. This hypothesis is that the eukaryotes evolved from within the archaea and not separately from each other. That is, this is the three-domain hypothesis, where each of the individual bacteria, archaea, and eukaryotes evolved from separate lineages. Okay, like in almost everything that we do in science, when somebody publishes a paper, somebody else says bullshit, right? That's just the normal way that science apparently works. Not only because that's the way science should work, you always want to question things and probe things, but it also turns out that a lot of scientists are assholes. So, in subsequent years, people said, I don't believe you. So we were able to actually get more genomic information from these so-called Asgard archaea. So we've got Thor archaea now, Odin archaeota, Heimdall archaeota. I'm waiting for Tony Starkiota. I'm sure that's going to come at some point, right? Scientists think they're clever. They're not. We're stupid.
Were these all named for the same group, or did they all go like, let's do this?
No. These are actually separate researchers doing this. Not that now. To be fair, they did isolate these fragments from, at least initially, from Loki's castle, this hydrothermal vent, right? So it made sense to kind of stick with this theme. But yeah, this is kind of crazy shit. But whatever, it's cool. But at the same time, other researchers went back and they wanted to look at the original data methods, right? The data set that was used to make these discoveries in this Nature paper. Okay, so this was published back in 2015, which is in terms of science not really all that long ago. All right, so now the authors are going to get the original data sets, different papers. There's a different scientific group that's going to obtain the original data sets and now start monkeying with it to try and figure out some stuff. All right, and when the authors took this data set and performed the analysis in exactly the same way that the original authors did, they get the same results, right? That is, the eukaryotes evolved from a branch related to Loki 3. Cool, I can repeat this science. That feels good. But now the authors are going to say, hey, you used proteins for this analysis instead of DNA, which is a little bit atypical of this. In addition, why did you choose this set of proteins? What happens if you choose a different set of proteins? All right, so now this is what we're going to do. We're going to use these so-called Woese proteins. All right, this is the same dude who's the senior author of the paper that you're reading. These proteins are so-called universal proteins. These are the proteins that all organisms have and utilize in order to survive. And of course, these are like DNA polymerases, RNA polymerases, and ribosomal proteins. There are 11 proteins that were picked for this procedure. So now these authors are going to use proteins, a different set of proteins, and run a very similar analysis. And fortunately, they get a very, very similar tree. However, the eukaryotes now no longer branch out of the Lokiarchaeota, but rather are a previous ancestor to perhaps the Tac superphylum, which would be shown in here, basically. All right? So by using a different set of proteins, and these proteins are thought to be proteins that everybody should have, we get a slightly different organization. In fact, if you go back to the original paper and you take out a single protein from their analysis, this is the so-called EF2 protein. This, again, is an elongation factor, if you'll remember, for translation. If you actually are ever interested in this and you go back and you read this Nature paper that describes these Lokiarchaeota, you'll see that there was something really bizarre about that EF2 protein from this Loki3 group. There was a bizarre insertion in the EF2 open reading frame that, one, didn't make a whole lot of sense, but also made it much more eukaryotic than it would normally be. And none of the other Lokiarchaeota had that insertion. So if you take out just that EF2 gene from the analysis, to recall, this is the original study that shows the eukaryotes are branching off of the Loki3 group. Now, if you remove EF2 from just one strain, you take EF2 out of Loki3, then the eukaryotes come out someplace else. Now, they still come out within the archaea in this analysis. However, they no longer come out as directly from the Lokiarchaeota. And furthermore, if you analyze just RNA polymerase in this manner, you get yet a different phylogenetic tree. In this case, if you measure just relatedness of RNA polymerase, we restore this so-called three-domain model. That is, we have the bacteria, the eukaryotes, and then the archaea forming their own domains of life, with the Lokiarchaeota still found in here. This particular analysis suggests that the Lokiarchaeota are actually closely related to the Euryarchaeota, so everything down here in green is the Euryarchaeota. Remember, this is the group that contains the phanogens. Whatever the case is, the analysis of the Lokiarchaeota still identifies a new phylum of archaea. And at least from this paper, the eukaryotes did not appear to directly evolve from the Lokiarchaeota. Okay. So here's sort of a part 2 summary. It turns out that evolutionary science, building phylogenetic trees, it apparently is easy to do. Anybody with a computer, a couple of fingers, and the right programs and a bunch of DNA sequences can build these phylogenetic trees. But if you slightly alter the input, you can also have dramatically different outputs. So what does that mean? I mean, that's problematic. And the problem is that you can't really ever test this stuff. In terms of sort of the evolution of microbes and cells, when we study them now, we're studying a snapshot, right? As we see it. We don't really know what happened 250 million years ago. We don't know what this is going to look like 250 million years in the future. So all we can do essentially is guess. What this second paper, this follow-up paper did not do, however, is disprove the fact that Loki archaeota actually have eukaryotic-like proteins. So one important part of this was that the authors actually went in to try to determine whether or not 18S ribosomal RNA sequences were detected. They were unable to do so. If you'll remember that 18S ribosomal RNA is the small RNA used by the ribosome by eukaryotic cells as opposed to prokaryotic cells. Although that second paper does argue that there is potential eukaryotic contamination somewhere. And for example, that EF2 sequence from the Loki 3 group. And in the past, I left this lecture at, this isn't easy. We'll probably never see these organisms or much less be able to work with them. Remember, these organisms are identified by taking a sample of ocean floor sediment 3,500 meters below the surface, and all we're doing is looking at DNA, right? So how in the hell are we ever going to isolate this organism, grow it, and do science on it? And I actually thought that that's where we were going to leave off. But I did some quick search